Colleagues, now we have our third presentation as per our proceeding. The third presentation is going to be on metagenomics, a tool for assessing plant health through soil health. This presentation will be done by Dr. Shankar Ganapathi Shanmugam, Assistant Research Professor, Institute of Genomics, Biocomputing and Biotechnology. Sorry, I hear some feedback. Assistant Research Professor, Institute for Genomics, Biocomputing and Biotechnology, Department of Plant and Soil Sciences, Mississippi State University, United States. And he is a scholarly researcher in the area of soil microbiology. Dr. Shanmugam's research program focuses on use of soil metagenomic tools to understand how soil microbial dynamics affect soil health and by extension, plant health. Specifically, his research team investigates below ground microbial community dynamics and how they affect above ground plant communities, as well as how this interaction affects soil health. Thus, his research outputs provide Mississippi growers with the tools to maintain soil health for sustainable agricultural productivity. Dr. Shanmugam currently have a joint teaching and research appointment with the Department of Plant and Soil Sciences and the Institute for Genomics, Biocomputing and Biotechnology at Mississippi State University. Please join me in welcoming Dr. Shanmugam to deliver the presentation on metagenomics, a tool for assessing plant health through soil health. Dr. Shanmugam, the floor is yours.
Thank you, Dr. Sarvanakumar. Thanks for that introduction, but you forgot to mention one important thing for the viewers. I had the privilege of being a batchmate of Dr. Sarvanakumar during our undergrad at Tamil Nadu Agriculture University. So with that, can you hear me, everyone? Okay.
Yes, dear. Yeah.
Okay, Sarvanakumar, I'm not being able to share the screen.
Yes, I'm sorry about that. Just I make you co-host.
That's fine.
I excited when I see you. Yeah.
Okay, thank you. Can everyone see now?
It's seeing the screen, yeah.
Give me 1 second here. Okay. Okay, my name is Shankar Ganapathi Shanmugam. I have a joint appointment here at Mississippi State University, and I'm a lead PA in soil microbial ecology lab here at MSU. So the primary focus, like Dr. Sarvanakumar mentioned, is looking at metagenomic tools to assess soil health. Before I start my presentation, I would like to thank Dr. Sarvanakumar and the organizers for this webinar, for this opportunity to share my research views. And I listened to Dr. Davide and Dr. Ravindran, and some of my presentation could be redundant, but I would like to make it as concise as possible because of the time constraint. So this will be a brief overview of what I'm going to present today. I'll give you a brief overview of soil metagenomics and the relevance of microbial diversity and soil health, some of the methods that we use for doing metagenomics and bioinformatics, and application of these concepts in different research areas. And I will also highlight some of the related projects that we are working on, along with if we have time, I will just go over the microbial ecology program that we have at Mississippi State University. Okay. I think Dr. Ravindran had a similar chart of the productivity. Here I'm showing corn grain yield in the United States over the years. If you could see, the y-axis is the grain yield in tons per year, and the x-axis is the years. As you could see, the yield was steadily increasing over the years, marking every landmark, like when we had open pollinated varieties and when hybrid corn was introduced, there was a big jump. And when the synthetic nitrogen fertilizer responsive varieties were when introduced, there was rather a big jump. But I would like for you to point out this area. When we had that big jump in productivity, there was also this fluctuation in the yield. These fluctuations compared to when we didn't have all these developments is pretty drastic. So what happens with that is sometimes the farmers, they cannot recover from the loss of the previous year. So of course there are a lot of factors that's involved in this erratic yield situation. But I would like to just point out some of the soil health related aspects that govern this principle of sustainable agriculture. That would be the focus of my presentation today. Okay, I study microbial ecology. So per se, I don't focus on one organism. I just look at the whole community, both bacteria and fungi in soil. So understand the nuances, understand their networks, understand how they interact. We study the soil health by understanding soil microbial ecology. Excuse me. So the microbial ecology is the study of the biota that inhabit the soil, their function, the processes that they mediate, and the effect of their activities on the characteristics of the soil and the growth of the health of the plant life. The microbial ecology is defined as both the structure and functional. It has two components, the structure and the function. The structure is more of what is present there, the diversity that's there, and which involves the community dynamics. You extract the whole genomic DNA and you look for what is present there. And the function determines all the functionally related aspects of these communities, like cycling the nutrients, structural components, decomposition, soil respiration, everything. So these two components are our area of research. Okay, we all know that the prokaryotes they have an incredible diversity with relatively little morphological distinction. So when it's difficult to tease them apart, especially in soil. We hardly characterized all the microbes present in the soil. A single gram of soil has close to like 40 million bacterial cells, and one study says there could be close to 9,000 to 10,000 different species in a gram of soil. But as of now, fewer than 1% of the known microbe has been cultured and characterized. This is because of the Great Plate Anomaly, which is you cannot grow them on petri plates. No, they cannot. No, you cannot sustain them on a petri plate or growing them in vivo. The ability to grow as colonies in a rich cultural medium like that is very limited for many of the soil microbial species. The reason could be they have different adaptations and they have different protections that are offered in the soil, which you cannot give in a lab condition. If you look at this small figure here, if you assume that a gram of soil has like 4,000 species, when you extract and plate them in a nutrient agar, you would end up with 40 species growing there, which is roughly 1 to 5%. So before the advent of DNA sequencing, scientists thought that was the diversity of the microbial communities in the soil, and that's how they reported it. So when the sequencing technology came into effect, everything changed. The belief system, the total belief system in microbial, especially soil microbial ecology changed, environmental microbial biology changed. So they were able to sequence the microbial communities, and based on the database that's available then and now, we know that there's a lot more out there compared to what was growing in a plate. So this would be the core of what we do in our lab. We are trying to see who are there in the soil and what are they doing. So we extract the total genomic DNA in soil and look for what's the diversity present. We use different techniques for that. Of course, each technique has its own advantage and disadvantage. Like PCR, GGG, which uses barcodes for soil microbial communities. The clone banks, we still do some, but I mean, it's almost obsolete. The microarray analysis, we don't do much of it, at least the homolog of the known genes. We do more of metagenomic analysis these days, which helps in listing almost all the existing genes there. But again, each method has its own limitations. For functional analysis, we do some real-time PCR, and we also are venturing into metatranscriptomics analysis to characterize most of the expressed genes present in the soil. Okay, so it's easy said than done. Unlike single organisms or group of pathogens that you focus on, you can sequence the whole genome to characterize both the all the functions that it can do. Here we are dealing with a humongous diversity. So practically it is impossible to sequence all the organisms. So the solution is pick for some reliable gene, like 16S ribosomal RNA gene, which is highly conserved, and just sequence that gene. Of course, it has some disadvantages, which I'm going to talk, but it has proven to be ideal so far for characterizing soil microbial diversity. This is ideal because it's highly conserved structurally, and it has variable regions and conserved regions. I'm going to describe that in this slide. So the whole length of the 16S gene is close to 1,500 base pairs. There is roughly like 25 copies of the genes in each bacteria. That's another drawback with this gene. When you start counting one gene as one organism, but still, this is the best out there, I would say. It folds in such a way that it's easy to design a primer set for this. So this is the structure of the 16S gene. This is the component of the thickness, small subunit of a prokaryotic ribosome, and for the fungi, it is 18S. Okay, this is the structure. You could see with a lot of variable regions, which are flanked by conserved regions in between. So this makes it makes this very marker, very unique marker. So that's why we call it a molecular marker. It's present universally. It has conserved regions. So these conserved regions would help in identifying these bacteria, and they will be present. You can be sure that it will be in all the bacteria, and you can design a PCR to target those regions. So between the conserved regions, you will have those variable regions which are specific to the specific bacteria. So based on that variable regions, you can differentiate each bacterial species. So this marker has a complete package for targeted metagenomics. So when we say diversity, excuse me, we study both the alpha and beta diversity of a community in any environment. So far, we mostly we work on soil samples, but we have also worked on fish mucus, on some plant roots as well. But anyway, the alpha diversity is a diversity within a particular area or ecosystem, and usually expressed as the number of species. They also call it species richness in that ecosystem. A beta diversity is comparing the diversity between the ecosystems. For example, if you compare different samples, that's the beta diversity, and within sample differences is called alpha diversity. For the gamma diversity, it's a measure of overall diversity within a large region, because we did some biogeography work where we study the gamma diversity as well. Okay, so soil metagenomics. What's metagenomics? Metagenomics is the study of the genetic material recovered directly from the environmental sample. It involves sequencing all the DNA extracted from a sample, followed by assembly of sequence reads, and mapping them to a different database, and estimate the diversity measurements. So here is a typical workflow that we follow in the lab. Dr. Davide, he actually had some good slides, so I'm just going to go over pretty quickly on this. What we do is we extract the total community DNA from the soil samples, purify it, amplify the 16S gene or the ITS gene, or the combination of both in some cases, and then we sequence it, and then we do a taxonomic classification, and then we do relative abundance and taxonomic estimates, and then we compare those operational taxonomic units. We create operational taxonomic units by matching that with the database, and then cluster them. So the OD analysis and clustering, we can infer the alpha and beta diversity there. So that's the who are there part. and for what they are doing, we do some potential function analysis. We don't do a full-fledged transcriptomics yet, but we do some functional potential analysis, like using the 16S chain and using platforms like PyCrust, MGRAST, and all those online tools to annotate some functional potential for these genes, and then to just get some details on the broad functional categories of these organisms in any sample, in a soil sample or any environmental sample. So the soil and plant relationship, I cannot overemphasize on this. The plant or soil root interface it's a dynamic region in which numerous biogeochemical processes takes place, and they are driven by the physical activity and the diversity of the chemicals released. We call them exudates by the plant's roots, and they are mediated by the soil microorganisms. So this is a very, very, very dynamic area in the plant root, where a lot of things are happening. This region actually controls a host of reactions regulating the carbon input into the system, which could be correlated to a sustainable plant growth. Okay, so that's a brief introduction of what we do. Now I want to jump on, some of the research that we did. I usually present this result. This is close to my heart. This was my PhD thesis. This is where I got pretty excited about how the soil and plant relationship unfolds in a real life situation. Here, I was looking at a sand dune chronosequence across Lake Michigan. If you could see in this picture, this is the aerial graph, okay, and this is Lake Michigan, and these are concentric sand dunes that are formed due to climatic events, due to the shrinkage of the Lake Michigan. These dunes got exposed at different time periods, and exposing the sand, and thereby acting more like a timestamp. So these are called chronosequences, and they were already dated. Actually, they did some carbon dating based on the exposure of the sand to the atmosphere, and then they dated the carbon material present in those sand dunes, and the age was ranging somewhere around 4,000 years as you get away from the lake. And when you get close to the lake, that's where you get the fresh exposed dunes or the younger dunes. So the age range was from like 100 years to 4,000 years. So the idea here was to look at like using these chronosequences as a proxy for soil development, and then see how the microbial diversity and community composition changed over time across these chronosequences. So I took soil samples from each of those dunes, and these ridges were like closely spaced at approximately 2.5 kilometers long, and then like 10 to 30 meters wide. So I took soil samples from all those dunes. So the idea was, we had the hypothesis that the soil bacterial communities will follow a pattern of change. that are associated with soil and ecosystem development. So what we thought was there will be a lot of overlap with the younger dunes, with the microbial communities, and when the dunes got older, you will have a distinct microbial community based on the complexity of the soil makeup. But we were proven wrong, actually. I will come there. So this is a picture of one of the dunes. This is the youngest dune when I sampled, but there is another dune now exposed. The lake is still shrinking. So this dune was close to 100 years old. Otherwise, this dune was exposed 100 years back. And what happens is, once the dune gets exposed, the plants start to colonize these dunes. So you could see some beach grasses and small shrubs getting established in these dunes. And this is the oldest dune that I sampled. You could see that there's very less understory vegetation there, and these dunes they got inhabited with hardwood trees like oaks and pines. So there was a big change in plant community composition along this chronosequence, starting from small grasses and bushes, and as the dunes got older, they were established with hardwood trees like pines and oaks. So it was interesting to see the above ground and below ground link. In this graph, if you can see, so this is an ordination plot. The way you read an ordination plot, luckily here we just had a one dimension solution, but you might have multi-dimension solutions. But in an ordination plot, for example, in this plot, this axis shows close to 78% of variability in that data. So that means this is the major axis, and each of these points represent close to like 1,200 or 3,000 different bacterial OTUs or bacterial species. As you could see, along the soil development, the biggest change was seen in the younger dunes compared to the older dunes. So you have to see the change across this axis. So the variability between this age and this age. So this is the youngest dune, and this is close to like 400 years or something. So the biggest change in bacterial community composition was seen between these younger dunes compared to the 400-year-old and the 4,000-year-old dune in terms of bacterial community composition. So when we correlated, so this is the similar ordination plot of the plant community composition. The youngest dunes had the yearly succession species, plant species like small shrubs, beach grasses, and small pine shrubs. And the late succession species were hardwood trees, like I mentioned, oaks, spruce, and then loblolly pines, and all the big hardwood trees. So if you can see, the bacterial community diversity difference was following the change in plant species community, which was very interesting to note. So to test this hypothesis further, I also collected the soil samples in winter. So this is Lake Michigan, and it gets pretty cold in the winter. The temperature can go up to minus 25 degrees centigrade in winter. I took some soil samples in winter, and I I compared it with the summer samples. To much of our surprise, I mean, there was not much difference. So this is the another ordination plot, which was giving us a two-dimensional solution. The axis 1 had showed 81% of variability in the data. The axis 2, which is the minor axis, which only showed like 8% of the variation in the data. So as you could see, so this is the youngest dune, and these are all the older dunes. So the bacterial community, the older dunes kind of clustered together, and the big differences along this axis was again seen between the youngest dunes. And the red color and the green color indicates summer and winter sample. So they were pretty close together. So that means there was much variability with the summer and winter sampling. So we did correlate our results with all the soil physical and chemical characteristics that we measured. So we were able to do some variance partitioning analysis because there are various factors or confounding factors that could affect bacterial community composition, and soil variables by far are the strongest in all the literature. You can see that. So we were able to partition that, and soil variables were the highest, were by far the highest influences. And the geographic distance. Sorry, I forgot to include a result here. We did have another corona sequence in Georgia, so we are measuring whether the geographic distance will have any effect on that. We did find some result from geographic distance having an influence on the bacterial community. But still, in spite of all this, there is still close to 50% of unexplained variation that's present in the soils, which we cannot attribute to anything. In spite of characterizing all the soil, plant variables, there is still some variation that we cannot account for. Okay. Okay. So we do community genomics, and we use metagenomics as a tool for studying the community genomics. We mostly do targeted metagenomics, focusing on specific genes like the 16S gene or the ITS for fungi. So the advantage of doing that is we can immediately link that to a function, and we can easily design the primers. And there is high depth of coverage in a complex community because there is a big database available to blast your results and get the results. And also there's a big functional annotation database available for the 16S genes. Some of the negative aspects of this method is you are only focusing on a single gene. So there's always a criticism whether is it representing the whole characteristic of an organism. So we started moving into shotgun metagenomics, and right now we are working on comparing the results. The shotgun metagenomics has some advantages. It's a broad brush. It's an unbiased survey. You just randomly pick sequences from a genome. You target random areas of a genome, and then you sequence it, and then you blast your results and you compare it with the database. The problem is interpretation, because you cannot ID the genes in the sequence obtained. There is no enough database, like Dr. David pointed out. There's it's building up, but right now there's not enough database to cater the whole diversity that we are trying to capture in our samples. But they have a very good application in the next generation sequencing. You'll be able to confidently say that it could be that species, but still you are relying on 97% sequence similarity. You can go to 19% sequence similarity, and that's where you get into problems like mismatches. Okay, so that was the molecular biology aspect of it. We also do a lot of bioinformatics in our lab. So that's another big area of research that we do, because when you're dealing with a complex community, like an environmental sample, a lot of time, the interpretation of your results is the difficult part. You can get the sequences, it's easy to sequence, but knowing what is there, and are we properly capturing the diversity? All these questions depend on what kind of algorithm that you use, what kind of metagenomic tool that, the bioinformatic tool that you use, what kind of piping that you use, all those matter. So that's another important area of research in our field. Okay, so here is a flow chart or a pipeline that we use in our lab. So it just depicts concisely, what are the steps that's involved in sequence analysis. We do a paradigm sequencing with Illumina MyC. After all the quality filtering, you cluster your sequences into operation taxonomic units, and then from that step, you can match your sequences with the database, classify the ODUs, create a taxonomic profile, or you can do all the alpha, beta diversity estimates, and also do some network analysis. So this is roughly a flow chart of the pipeline that we use in our lab for sequence analysis. Right now we use different pipelines for data analysis. One of them is Mother, and another one is Chime 2. Both of them are open source softwares that are available free. And when it is open source, that means it has its own issues. There will be a lot of parameterization that will be involved, a lot of decision making that will be involved when you are analyzing your sequence. Okay. So here is a brief overview of the bioinformatics that we do in our lab. You do all the data. There are three major categories, which is data transformation analysis, visualization tools, and also the statistical analysis. So after you do all the ODU-based estimations like alpha, beta, diversity estimates, you start visualizing them. Like you can use bar plots, diffraction curves, hierarchical cluster analysis, and ordination techniques where because you're dealing with the multivariate data here, each organism is it has to be kept separate. So just imagine you're compiling or compressing your univariate data into one two-dimensional, three-dimensional solution. So we use multivariate ordination techniques for that. And then for statistical analysis, we use ANOSIM, Termanova, and all those multivariate analysis techniques to compare the results. So this is a brief overview of our lab. So to do this, you need a lot of computer power because I'm dealing with the core community analysis. On top of it, you'll have more samples. Usually, in a standard project, we'll have 100 to 200 samples. You have to run them simultaneously to create a distance matrix, and a lot of most of the time these distance matrix will be copied in your random access memory, and it will be processed there. So your typical laptop or a desktop, it cannot handle that much data. So we use high performance computing to handle that, supercomputers. We have a state-of-art facility in our university with close to 72 terabyte of main memory and 26 terabytes of RAM available. So we submit our jobs in clusters remotely, and then run the jobs in the clusters and get them back, and then work on those sequences. So the high performance collaboratory here at Mississippi State, it ranks 4th in the world in terms of university owned computing facility. So we are making most use of it. I think Dr. David pointed out about how cheap sequencing costs are getting. So this is the table from NIH, and if you could see, it started for the whole genome project. It started somewhere around 100,000, or I think it's more than 100,000 there. And today you can sequence the whole genome for like $1,000. So you can see that there was a real dip in 2006 and 2007 when the next generation sequencing technique kicked in. And from then, it's going down phase. But what does it mean? The real cost of sequencing is changing. I like this picture. It's from Genome-Biology. It was published in 2011. And if you could see, so the experimental design, then comes the sequencing, then you do the data management, like analysis, like data reduction, mapping, all those bioinformatic tools that you use. And then comes downstream analysis, where you actually study the various interactions, various expressions, and various functional annotations and everything. So if you can see how this is evolving, this is pre-NGS, next generation sequencing, which is approximately around 2000. So the biggest money was put to sequencing. So the whole chunk went to sequencing. They call it now, which is approximately 2010, still half and half. But they're calling it future. We are already here. It's 2020. Most of your bucks goes here, like how you interpret the downstream analysis. Sequencing is getting cheaper. And another area that is gaining importance is experimental design. How do you design your experiments? What are all your questions? What are all the important questions that you have? What's your hypothesis? Those are getting more focus than sequencing per se when it comes to any sequencing projects. So sequencing, having a big data, it comes with its complexities. Most of you know about this. And it gets even complex when you're dealing with environmental samples, when you're dealing with metagenomics, you're dealing with all the organisms. We're trying to picture what kind of change is going on within the organism, within the population, between the population, within the community, and between the community. For example, here is a network analysis from a study, and you can see the different phyla of bacteria, and you can see each line or node denotes the relationship between two organisms or two phyla. In this case, they are designated as phyla, and the bigger the circle indicates larger, the richer the community is, and closer they are, that means they are closely related. So this is the kind of complexity that you're dealing with. So how do you address that complexity is trying to separate the factors, do some variance partitioning, and also look for patterns. For example, look for generalists, broad categories like generalists and specialists. And in soil microbial ecology, you can call a community as a generalist when it is when it can use all type of carbon sources. Specialists are organisms that can actually work on recalcitrant carbon. That's an example. So if you can separate your communities based on that, then you can tease out this complexity to some extent. Okay, so that's the overall picture of soil microbial ecology. So with some slides that I'm going to talk now, I'm going to address how we applied this for different projects. Right now we are working on a disease suppressive soil project, and we are looking at how the native microbial community in the soil can help in suppressing a specific pathogen. Okay, we are working on a taproot decline pathogen, and looking at how the natural microbial diversity in the soil can change, or how it can suppress these pathogens. So we address this suppression into two broad areas. There is a general suppression and there is a specific suppression. General suppression is the ability of the soil to inhibit the soil pathogen due to the collective competitiveness of all the organisms that's put together, and they create an antagonistic activity against a pathogen. Specific suppression, in contrast to the general suppression, it deals with specific microbial species which could be dominant in the community, which could act on the pathogen. So a lot of time, the specific suppression, it could be an induced suppression. So for example, all the biocontrol agents that you use, so that's the theory that they try to exploit when you are using a biocontrol agent. but I'm interested in studying what's the percentage of suppression between these two phenomena in soils. So this is VCP Soybean Promotion Board funded project where we are looking at this soybean taproot decline and how the native microbial community affects this pathogen. So we are looking at the overall microbial diversity. We are also doing some quantitative PCR on the targeting the xylaria, which is the causal organism of this disease. So we did some field rating of this disease, and then we obtained rhizosphere soil samples, and we're still looking at the results. But the idea here is look at the gene copy numbers of those pathogens and how the crop management affects the disease intensity and the community structure, and do some network analysis to tease out the major factors that help in the suppressing of the soil. We are also doing some functional diversity estimates to see which factor has a major influence on the functional diversity of these organisms. So quickly here, here is another study. So this is a study that we did. We already published this. We looked at different tillage intensities to see how tillage intensity affects soil microbial diversity. We got some interesting results. Here is another ordination plot. After like 3 or 4 years of this experiment, we had 4 different tillage intensity: minimum, moderate, high, and reduced. See that the minimum intensity tillage treatment has had a very significantly different microbial community composition compared to the conservation, the conventionally tilled plots. So this is just after like 3 years. The first and second year we didn't have much difference, but the third and fourth year we had a very significant difference. We also did a functional analysis on those soil samples, and we could see there was some significant difference in the carbohydrate metabolism between the conservation tillage and the conventional tillage plots. So this is another project that we are working on right now. We are actually in the process of developing this. We are looking at the cereal rye cover crop as a modifier of soil microbiome function. So we are just trying to characterize the functional diversity of the cereal rye associated rice microbiome. We are trying to figure out how the root exudates from the cereal rye plays in selecting the microorganisms around the rhizosphere. We are trying to do some metabolomics and metatranscriptomics work there to see what kind of root exudates are there in those systems and what kind of organisms that they are trying to recruit. So this is a picture. I just wanted to show this am I good on time here? We are trying to show that the microorganisms have a very good signal systems with the root exudates. So that's what we are trying to find out in this study, to see what kind of microorganisms are recruited and how they are recruited and what kind of genes are upregulated both in the plant roots and in the microbes. Okay, so overall, this is an interesting publication. This is from Nature. How can soil biodiversity affect human health? So this is just an interesting cycle that they put out there. The soil microbial diversity, the species richness, the functional diversity. If you have the loss of that, so that means you are using an ecosystem functioning there. For example, water infiltration, regulation of pest pathogens, erosion control, everything might go down. So that might significantly increase the soil-borne pathogen or reduce the supply of food and clean water and air to the system. So that will directly affect the human health. So this again counteracts with the climate change, poor land management again, that comes back to soil biodiversity. So that's a big cycle. It's interesting to note how soil microbial diversity could have, this is not direct, but still there is an indirect connection with the human health. So finally, this is the project. So wow, how ironic. COVID-19. So we are working on a COVID-19 project. We are actually developing this project. What we are trying to do here is to see the presence and the ecology of the COVID-2 virus on an on-site wastewater system in the Mississippi Gulf Coast catchment area. So what we are trying to do is collect municipal wastewater from both hospital systems and rural areas, and then we are going to compare and see if we can develop an early detection system of the COVID-19 outbreak using the wastewater system. So there we are trying to capture the COVID-2 load and also the natural microbial communities. Are they interacting with these virus in those wastewater systems? Are they providing nutrient to those viruses? What is the dynamics? So I hope we'll get some interesting results. There's already some result published, but we are trying to extend that. So with that, I think I'm close to my time. This is my lab, and here are our lab members, Dr. Kingiri, and I have a postdoc, Nisarga, Amita, Sapna, everyone. So we as a group, most of the results that I showed you are work of these people. And this is just some of the highlights of our program here at MSU. I think I'm close to my time, so I'm just going to finish up here with this slide. This is our football stadium, jam-packed. It was not this year. This year, it was just a 25% occupancy. So I hope things will change next year. On that positive note, I would like to thank again Dr. Shanmugam and the organizers for this webinar, to giving me a chance to present. And if you have any questions, I'd be happy to entertain any questions. Thank you.
Thank you, Dr. Shanmugam, for this excellent and very informative presentation on soil microbiome and biodiversity and how that could affect plant health as well as the soil health. So, colleagues, if you have any questions to Dr. Shanmugam, and he would be ready to take those questions. I think our colleagues and participants are a little shy, it seems, to ask the questions.
Maybe they are tired.
Should be. Actually, I thought of having a break, but I thought it would be good for face-to-face in a virtual platform. I thought, it's better to go to one shot. Yeah. So.
Are you guys having a break? It should be close to lunch for you guys.
Yeah, because of a virtual thing, I thought, everyone would be having a virtual lunch. So, dear participants, if you have any questions, please post that in chat box, or else feel free to raise your hands, and so that you can ask the question directly to Dr. Shanmugam. Okay, if there are no questions, I just have one. You spoke about metagenomics, right? And what could be the challenges in case of transcriptomics with respect to databases, as well as suppose in some cases we do not have the databases, right? In such instances, what could be done and how we can take it forward?
Well, that's a good question. We don't know because a lot of this depends on the previously existing database. Of course, you can, as a team, you can develop some database, but still, we're talking about big diversity here, and the expression genes for each of those species or even a subspecies, that there will be big variability there. So, so what we are trying to target here is a trend or a community. I'm just looking for a better word here. Just see the overall functional annotations. You cannot go for very specific functions there. Of course, there are like close to like 25, 60 functional groups that you can broadly categorize, like carbon metabolism, nitrogen metabolism, phosphorus, whatnot. But again, you have to take that with a pinch of salt because it's only from a limited database. Just like you sequence one genome at a time, these things, as scientists work on those, they just develop a database, and then that will make the results more and more robust. But with the present database, you can still address sample level differences with broad functional groupings. It's still possible.
Okay, thank you. So I see a question in chat box. I will read the question. The question is from Mena Bernard. How can the results obtained from soil metagenomic analysis be applied to enhance plant health and yield of crops, since we cannot culture most of these organisms, although we know the functions of the genes they possesses?
Well, here again, it's just it will be based on the correlations. For example, the disease suppression study that we are doing. So we have to. So you can go close to the rhizosphere, and then the assumption there is that you are capturing the diversity that is very closely related to the plant. Again, you have to go and characterize the endophytes in the roots, and then see how they can relate to the rhizosphere communities. So this is the game of correlation. So you know that there is a connection, of course. So if you look at a specific plant pathogen, for example, the xylaria, the taproot decline, if you could take soil samples from the rhizosphere of plants that are exhibiting different intensity of the disease, and then if you can do a correlation between those intensity and the pathogen count in the soil, you can do a correlation and then say they are correlated.
Okay, thank you. And also I understand that once we have the correlation, probably the soil interventions can be made to enhance that particular microbial population so as to protect the
pathogens. Yeah, that's what we are studying as well, like tillage or cover crops or any kind of management, even pesticides. So what are a combination of all these practices, how it can push the microbial community to the direction where we want it to go.
Okay, thank you. Also, if I understood correctly from your presentation, then based on the transcriptomics, and if we are able to tag the genes or proteins, and then we will be able to identify the genes based on it, and probably it could be a source for a genetic modified plant.
There are stable isotope probing where they isotope probing for seeing what kind of organism enter into the plant, which is closely related. But that's one organism at a time. So yeah, it's still possible. Yeah.
Okay, thank you very much, Dr.
Thank you
Shanmugam.
for having me.
I know it's your lunch time, and hope you will be able to join us around 2 o'clock our local time. And but if at all we have any questions, then we can have a discussion during the time. Otherwise, also still we can have a discussion, and probably someone also would be interested in collaborating your project, or else the students will also be interested, because we have some of our graduate students here, and they would be interested in knowing more
Sure.
about your research, and would be interested in collaborating with you.
Sure.
Right?
Sure.
Okay. Thank you, dear friend. Yeah.
I'm going to stay for your presentation though.
Oh, great. Thank you.